Good afternoon, everybody. Thanks for turning up. It's my great pleasure to introduce you, Mr. Manjappa. He's registered at University of Agriculture Sciences, Bangalore, GKVK campus, in Department of Genetics and Plant Breeding, and his research work is on finger millet. It's another great opportunity for us to have his presentation for two reasons. One is that in recent past, during our governing board meeting in mid-September, finger millet was added to our mandate crop list. So this gives us a better, I would say, visualization and prominence to raise the finger millet profile. And second one is this is actually celebrating our very productive collaboration with All India Coordinated Research Program on Small Millets, based again at GKVK campus in Bangalore. And more importantly, the interactions with University of Agriculture Sciences Bangalore to host the students. Taking these two, I would really like to thank two important guests. One is Dr. M.V.C. Gowda. He's really a, let's say, he's pioneer for finger millet research work and crop improvement in India. And I really appreciate your work. Without that, things would have never moved to this level. He's just retired retired couple of months back, but interesting, he's still keeping his interest in finger millet. He just informed us that he's already shortlisted now accepted offer as emeritus professor to work on finger millet at GKVK. So, sir, we look forward for that interaction to take this to new level. We know that we would need your expertise. Till that time, you are interested. So thanks very much for making to this seminar. And second one is Dr. Rangaya, Dr. Manjappa's supervisor, head of the professor and head of the department at Genetics Plant Breeding Department, University of Agriculture Sciences, Bangalore. Again, thank you very much, sir, for making it at short notice. We really appreciate your presence. And taking to Manjappa, he has spent us almost 6 months now, 7 months. And this interaction again started with M.V.C. Gowda's initiative to take some molecular work. So we have been helping. He has spent his time working on some of his marker data analysis, designing SNPs, identifying SSRs for partial male sterility, doing some diversity analysis, all of that. One information more about his work is this is being a really kind of auspicious occasion for us, knowing that finger millet, it would be one of the first seminar talking on finger millet. We requested Manjappa and also Dr. Rangaya to give us a permission to present his complete research work. Otherwise, usually this kind of seminar we restrict only to the part of research work a student or a fellow or visiting scientist who does part of his research at ICRISAT. So thank you for that permission, and he will be presenting all of his research work. So it would be a really nice opportunity for us to see what is being happening in finger millet in India. So nothing more, not much taking time further. I request Manjappa. So I welcome you, and its floor is yours.
Thank you very much, sir. Very good afternoon, everyone. Today I am presenting my PhD research work on heterotic potential of hybrids based on partial maize sterile line and development of maize sterile lines with seedling marker in finger millet. Coming to the introduction of the crop, finger millet, which is grown mainly in semi-arid tropics and especially in rain-fed condition. And it is growing more than 25 countries of Asian and African continent, and it is known for its nutrient rich profile like crude fiber content, calcium content and iron content, and it is also having a major like health benefits, especially for diabetic patients like hypoglycemic, cholesterolemic and anti-ulcerative. These are the properties. And coming to the decade-wise area production productivity of the finger millet over last 6 decades, we can see here from last 3 decades, the area and production is declining phase due to the replacement by other competitive crops which is having higher productivity, where research program has been taken place at a high level. So in that context, if you see the productivity of the finger millet, it is reaching stagnation even with the current breeding efforts. So in that context, heterotic potential of finger millet, the exploitation of that heterosis is important in finger millet to break the yield barriers in finger millet. Coming to the crossing techniques in finger millet, as we all know, it is highly self-pollinated crops owing to these two properties like cleistogamous and chasmogamous. It is depends on the climatic conditions. These two mechanisms works. Here you can see here in finger millet, especially at anthesis stage, the both anthers and stigma will come outside and the anthers will burst outside. So this is the mechanism of selfing. So we can also exploit this mechanism for increasing the outcrossing here. If you use the male sterile lines, in that condition, this mechanism can be exploited. When you see the artificial, if you want to go for artificial emasculation, it is very difficult in this crop because of very tiny flower. We cannot handle the flower with bare eye. So it is very difficult in this crop for hand emasculation. So the only method is contact method where we are following to hybridize the finger millet. So some of the breeding hybridization like heterosis work has been taken place in finger millet. But the problem is they have restricted the parents to pigmented types and low hybrid recovery. So in that context, for facilitating the hybridization in finger millet, exploitation of genetic sterility is significant. So, but in GMS, in complete GMS, the problem is the maintenance. Every generation we have to maintain. So, project coordinating unit, small millets, Bangalore has developed one genetic resource called partial maize sterile mutant that is called as PS1, which has been developed under the background of GPU28, which is a very famous variety in Karnataka, which is high yielding and which is resistant to the major disease called as blast, like finger blast and neck blast. It is resistant, and it can be maintained very easily. Just by putting a bag or growing in isolation, we can maintain the sterile lines. That is the importance of this line. And it is over season, it has shown a stable performance. When you look at the seed set percentage, we can see here in closed condition it is 11%. When you allow for open pollination, it is 20%. When you look at the progeny, the open in closed condition it is given true breeding. In open pollination, the progeny consists of both sterile and fertile. It means that there is a possibility of outcrossing. Outcrossing has been taken place here as compared to GPU28. When you look at the extent of outcrossing in finger millet, we can see here we have used PS1, which has been derived from GPU28. We have used both partial line and partial maize sterile line and its wild parent, fertile version GPU28. We can see here the hybrid recovery is 76% as compared to 4 when you use fertile into fertile grass. Likewise, the range was from 45% to 76% as compared to 4% in fertile into fertile grasses. With this background, my objectives has been framed. 4 objectives: characterization and inheritance of partial maize sterile mutant PS1, and characterization and inheritance of virescent mutant. This is a leaf chlorophyll mutant which has been isolated in GKVK Bangalore millet scheme, and assessment of heterotic potential of hybrids with partial maize sterile mutant, and development and evaluation of new partial maize sterile lines with and without seedling marker. Coming to the first objective, in our material we have selected 5 crosses to check the percent seed set in the crosses. If you can see in this picture, when we cross with fertile version, this is the female parent, this is the female parent after crossing. You can see here seed set is very high. In this VL315, this is one cross. It was ranging from 27% to the 49%. Even there is a scope for improvement in seed set as compared to sterile plant which is 9% in this material, and it is 83% in fertile version. When we look at the percent hybrid recovery in our material, we have crossed PS1 with 46 parents for the study of heterosis, which will be dealt in future slides. So using that material, we have we have estimated the hybrid recovery, which is range from 28% to the 81% with the average of 56%. We wanted to see what is the mechanism happening in the partial sterility, why it is partial sterile. So in order to understand that mechanism, we were start to study on anther-dehiscence. Whether is there any defect in anther-dehiscence? So we wanted to study that under microscope, just binocular microscope, we are seeing in the mutant anthers. We find that there is a normal dehiscence. Anther-dehiscence, there was a bursting of the pollens as compared to wild type. So there was no defect in that. After that, we wanted to study pollen viability test by using both strain, like acetocarmine strain and iodine solution. So in both the cases, you can see here in wild type and mutant type, we found that both have 97% of pollen fertility. That means there was no defect in pollen fertility by using staining methods. Then we move to the in vivo pollen germination on the stigma itself in the field condition. We have collected the flower from field in the early morning, that is the time of anthesis. We have collected and we have seen the wild type like GP28. Wild 28 means it is self flower we have collected. We found that after staining with aniline blue, which is a fluorescent dye, we could able to see many of the pollens would able to set its pollen, and we would able to germinate its pollen, and it could able to pass its pollen to the ovule, and thus it has produced some normal seed set. When you look at the partial sterile line, only few pollens able to germinate, but they could not set its pollen or pass its pollen tube to the ovule. You can see here, it has arrested here. So there was a defect in pollen germination. But when we see, in very rare cases, we found some stigma having a pollen germinated its pollen tool and reaches ovule, and thus it has produced some seeds. This is the mechanism why the partial male sterile has set some seeds and it has shown 90% of sterility. We wanted to see the inheritance pattern of male sterility by using 40 F2 population. Again, these material are from 46 F1 developed for heterosis study. We have used that, and we have found two different kind of segregation ratio, like 15:1 and 16:6, which like three genes and 2 genes kind of segregation. Like 11 crosses were shown this 15:1 and 29 crosses shown 63:1. We were confused. Again, we gone to F3 population, where we have selected 55 fertile plants in F2 generation of one cross. We have raised in F3 families. We have looked for the segregation of that fertility in the family. One interesting thing we got, like 38 families have shown fertile segregating. Means they are segregating for sterility, but 17 were they were not segregated. They are homozygous, fertile homozygous, but ratio is 2:1, which is significant. So which is fitted into 2:1 ratio, which is like Mendelian ratio, which is like 2:1 ratio. It concludes that it is governed by single recessive gene by using this ratio. And we can also give some idea why there is a low proportion of sterile plants in F2 population, which may be due to the gametic selection happened during anthesis time. Coming to the characterization and inheritance study of virescent mutant, it is a chlorophyll deficient mutant, which has been developed at ICRISAT, Bangalore. This is like yellowish green color because of deficiency of the chlorophyll. and it has shown its expression from first stage itself, like first leaf stage itself, till tillering stage. After it reaching tillering stage, it has passed to the grand growth stage. During that, it has started converting into green type. And from this stage onwards, it has converted gradually and it has become green, less like a white fly. This is that chlorophyll content of mutant and white type in during different growth stages. You can see here during early stage like seedling stage, the in white type, the chlorophyll content is double than the mutant type. Okay. When you look at the tillering stage, after tillering stage, there is a rapid increase in the chlorophyll content. You can see here. Because of that, we have seen green reversion in this stage. And when it starts advancing its growth stages, it tends to increase its leaf chlorophyll content towards white type, and it looks like a chlorophyll. It's like white type. And we have also identified that it has shown stable performance, like stable expression over seasons. We have studied for several seasons, and it has shown green revertible nature, and it has shown temperature insensitiveness at seedling stage. When we grow the seedling at high temperature or low temperature, it has shown same expression. But if you see at the temperature sensitivity for green reversible nature, if there is in a high temperature, like during summer season, the conversion rate is very fast. Like the conversion from yellow to green is very fast as compared to cool time, like low temperature time season. When you look at the agronomic performance of the mutant, the chlorophyll deficient mutant, the mutant has delayed flowering and having reduced plant height as compared to wild type, and remaining characters it is having same performance as compared to GP20 as compared to wild type. And this is the 16 backcross populations we have used, where we have categorized green into green and virescent group. Here we can see virescent having a delayed maturity compared to green types in all the crosses. When you come to the inheritance pattern of the virescence leaf color in mutant, we have studied 7 different populations of huge populations, like 1,200 or 2,000 till 2,000. We have taken the population size, and all populations are fitted into 3:1 ratio. It means that this trait is controlled by single recessive gene, which has been validated in every population also. Like here it has fitted in 2:1 ratio as like Mendelian. So what are the utility of virescent marker? Means the chlorophyll deficient mutant. In finger millet, as I already told, identification of F1 is very difficult without any marker. So this is a robust marker. At seedling stage, even at first leaf stage only, we can identify which is true F1 or self-1. So it will help to identify, and it can save time and resources, and even it will help the breeder to handle more number of material, especially in finger millet. He can handle as many number as possible by using this marker. And we have used this marker to determine the percent F1 recovery in contact method of crossing. As I told, contact method is the only famous method in finger millet for crossing because of easiness. So in this crossing method, earlier we don't have the data like what is the recovery, hybrid recovery. So we have used this marker and we have identified recovery percentage. It was ranged from 0.2% to 15% with the average of 5%. As I told, virescent marker is a robust marker which will help in crossing program in finger millet. So to facilitate hybridization breeding in finger millet, we have developed a diverse kind of virescent lines. So which can facilitate like early groups, late groups, tall, dwarf, and different like yield related traits. So different kinds of diverse virescent lines we have developed, and we have evaluated under augmented design using 4 checks. For all the characters under study, there was a significant variation, and this is the range. Like we had blast leaf resistant lines, early lines, and dwarf lines, and especially for grain yield, we can see some recent lines have superior yield than the checks, which is GP20. It is a famous variety. Some lines have higher yield than the checks. We have used top cross method to test the superiority of the virescent lines. You can see here we have operated like thousands of crosses we have made to identify a superior virescent line in the by looking at its progeny performance. We have selected based after seed set, we have selected 33 crosses, 33 plants which are having superior characters like yield related characters like finger width or something. We have used INDAF9 as a tester, which is a good tester. Yeah, based on its progeny performance like heterosis percentage and breeding value. Considering these parameters, we have selected different virescent lines for different characters. Coming to the assessment of heterotic potential of hybrids using partial master mutant. We have used 46 parents, which consist of 28 varieties released from different parts of country. Like star indicates the varieties and diamond shape indicates the germplasm. We have collected germplasm from Eastern Africa and India. They are elite germplasm having different superior characters like calcium or tolerance to drought, salinity. So different kinds of varieties germplasm we have selected for the study. We have followed contact method of crossing to develop the hybrids. Yeah, each hybrids were raised in 3-meter length of 3 rows in 2 replications by following normal agronomic practices. And we have followed RBD design to evaluate the hybrids in 2 replications, and we have evaluated in 2 seasons, summer and kharif, to check the stability and specificity of the hybrids over the season. We have followed restricted randomization to avoid the competition between early type and late type. This is the field view of these are all the hybrids evaluated in 2 replication during summer season. This is the pooled ANOVA from both during both season. You can see in the parents hybrids and parents versus hybrids for all the characters under the study, we can see the variation, significant variations we can see. Coming to the mean and range of heterosis of 46 hybrids for yield and yield attributing characters, we can see here we have earliness, like earliness to late type, and here also like early hybrids we got. Likewise for test weight, we can see hybrids with 13% of heterosis in summer season and 11% of heterosis during kharif season. For yield especially, we can see up to 58% of the heterosis we got during summer season and 48% during the kharif season. These are the top 3 hybrids selected based on these 3 important characters. These are the early hybrids having very good hybrid heterosis along with their means. So you can see these mean values are lesser than the check. That means they are earlier than the check. And like looking to the 1000 grain weight, these 3 hybrids have shown equally in both the seasons, equal performance in both the seasons, having higher test weight than the parents. Check. When you look at the grain yield, these are the hybrids which have shown high heterosis during kharif season and during summer season. So this is the hybrid which has shown good heterosis in both the season. That means it has performed very good in both the seasons. So it is a very potential hybrid. So you can see here this hybrid has 20 and 22 gram of grain yield per plant as compared to the checked 16. Yeah, based on this hybrid data, we have calculated like breeding values from all by using top cross method of analysis. We have analyzed the breeding value. Based on breeding value, we have selected the parents. These are the top parents which are having very high breeding value for different characters. So these parents will be utilized for the breeding program to improve the crop for the crop improvement, since they have the very good breeding value. When you look at the genetic parameters for different characters like flowering, tillers, and plant height, flag leaf, finger length, finger number, test weight, for these characters there was a correlation like association between parents and its progeny. It means that we can select the parents based on their parental mean performance. So it will be this data will be helpful in selection program. This is the regression of breeding value to its phenotypic value. That means it is analogous to the heritability as per the J.R. Sharma book. They have mentioned this. So using this data, these are the characters which have shown significant regression value for plant height. For yield, grain yield, since it is a complex character, it has shown very less heritability. So it by using this test, we have found less heritability. Coming to the parental diversity analysis based on phenotypic and SSR relic data. Yeah, we have observed 18 characters. By using 18 characters in parents, 46 parents, we have analyzed principal coordinate analysis using Darwin software to see the distribution of the parents. By using Gore's method, we have calculated the distance, and we can see the wide variation distribution of the parents in both the seasons. This means that the current genotypes have wider variability. When you look at the clusters are based on geographical origin of the genotypes. You can see here, suppose this is a purple line which is from Karnataka. So like this, the clusters have shown it is shown like the clusters are based on their origin. The lines from same origin clustered in the same clusters. Some exceptions are there. For the genetic diversity of parents, we have used 16 SSR marker to screen the genetic diversity. This is the summary statistics of the markers which I have used. The peak percentage was from 2% to 61% we have got, and this is the marker which is having high peak content and gene diversity. When you look at the phenotypic data and genotypic data, like we have used Mantle test to see the association between the matrices derived from phenotypic data and genotypic data. So we have tested that. Here we can see phenotypic data based during summer season, phenotypic data during some kharif season, both have correlated. It means that these parents can show same performance in both the seasons. That means that we can sow the crop in off season, you can evaluate. So this is the data, information we can get from this correlation. And when you look at the phenotypic data and versus SSR data, we found correlation, less correlation. This is the contribution of, this is the F statistics where you can see the contribution of variety as a group and germplasm as a group. Within germplasm, this has contributed 37%. the variety group has contributed 55% to the total variability. This is the principal coordinate analysis distribution graph, which has shown based on SSR leak data. We have used two axes where each has contributed both axes contributed 32% to the variability. We can see the distribution of parental lines on the two axes. When you see the correlation between genetic distance and heterosis, if you want to see the associationship between the genetic diversity of the parents and heterosis in the hybrids, we found that the correlation is only observed in plant height and flag leaf width in summer season. This is the respective regression values. During Kharif season, we have seen four characters like peduncle length, flag leaf width, and test weight. These four have shown co-associationship between parental divergence and heterosis. Based on the correlation based on SSR data, like distance based on SSR data versus heterosis, we could not find any correlation between them. When you look at the heterosis, we have clustered the different genotypes into 4 groups here based on the distance, and we have find the average heterosis for each of the groups separately. For each character, you can see in the first group, which is containing GP28, which is a female parent for all the hybrids. The cluster containing both male and female parent having very less heterosis. Yellow indicates less heterosis and orange indicates high heterosis. So low heterosis is found in intra-clusters. That means that higher the distance, heterosis will be higher. Likewise, during Kharif also we have found similar results, like cluster containing GP28 having lower heterosis for many of the character. Likewise, in group 2 also we found, since it is group 2 is related to or near related to group 1. Based on SSR leak data, we have clustered the genotypes into 6 clusters, where we can see here, as I told, PS1 is derived from GP28. That's why both are comes under same group. As I already told, G1, which is a donor for virescent character, this is a mutant from GP end of 8. Both are coming under same group. Yeah, based on molecular data, distance data versus heterosis, wanted to see the relationship. You can see in the cluster 5, which is containing GPU28. The cluster containing GPU28 having lowest heterosis. I mean to say that intra-cluster process having less heterosis as compared to other clusters. Likewise, for summer data also, 4th, 5th and 6th having lowest heterosis. I mean to say that distant clusters have higher heterosis for different characters. Coming to the development and evaluation of partial sterile lines with and without seedling marker. As I already told, seedling marker is a robust marker. We can identify a F1s at seedling stage only. We have already a partial male sterile mutant. If you combine this marker, like virescent marker, to the seedling male sterile line, we can identify the self seeds or self plants and the nursery stage itself. So that we wanted to do. So we have developed different diverse male sterile lines. Like we have selected 150 partial male sterile lines derived from 46 hybrids, which were used for heterosis study. Based on those 46 hybrids, we have raised in F2 and we have selected sterile lines, and which were evaluated in last season, and we found that there was a wider distribution of sterile lines which we have selected. These are the range of these lines which are having early flowering and dwarf lines, and high finger number, high finger length, and productive tiller number. For these characters, we have very good sterile lines. These are the sterile lines, diverse sterile lines having peculiar or specific characters. Like you can see here, this is having very good finger width. This is the partial sterile line. This is the sterile lines having longer fingers. This is the line which is a combination of both sterility and virescence, and also it is dwarf. This is the line which is having larger seed size, means higher test weight. This is the line having open ear shape, which is preferable in finger millet. And naked type, this is having naked type. This can also be used in some analysis or some study. As I already told, combining virescent character to the sterile background will help in identification of F1s at seedling stage itself. So we have demonstrated that technology using three hybrids. By using contact method of crossing, we have developed three hybrids. We have raised in nursery. The nursery consists of both self plants and crossed plants. The self are virescent and crossed ones are green. That means that green is a dominant character over virescent plant. And in nursery we have selected, or we have isolated two hybrids, and we have transplanted in main plant. And ragi is only a crop which can be transplantable. In three replications we have evaluated the hybrids along with some checks, and in these three hybrids we find very good hybrid here. That means that we can say we can use this female parent for further study or further hybrid development. This is the SNP pipeline. We have sequenced PS1 and GP28 by using DDRAD sequencing method. We have used Illumina ICRISAT 2000 using ECOR1 and MSC1 by double digestion. Initially we got 80,000 base calls, and by using different criteria, filtering criteria, we come out with 68 SNPs, and we have used 68. We have used 68 SNP for online blasting to screen out or to see the functional annotation, and two out of these 68, two have non-synonymous SNPs. We have rejected these, and 27 clusters have not hitted in the online blast, and the remaining 39 have hitted in the online blast. We have made the functional annotation for them. Coming to the conclusion of my PhD research work, we have already seen for many of the characters, including yield and its contributing characters, we have seen a very high heterosis for all the characters. So for grain yield per se, these are the four hybrids which have shown superior performance over the check, which is GP28. Over that, we have very good hybrids, especially first hybrid. This hybrid has shown a stable heterosis over both the seasons. So these hybrids can be used for further recombination breeding program to isolate a superior, to get a superior recombinant lines or superior segregants in the future crop improvement method. And this finger millet is a highly self-pollinated and allopolyploid crop, where epistatic component plays a major role in controlling a complex characters like yield. So in order to have a desirable expression of a particular character, we should have a desirable combination of a gene. So in order to get desirable combination, we have to there should be a shuffling of the genes to get that desirable combination. To get the shuffling of the genes, we should frequently cross the material to get the desirable combination. In this crop, crossing was difficult. By using PS1, which is a sterile line, we can cross repeatedly to get the superior combination of the genes, so that we can improve the yield or any complex character. That can be done using male sterility facilitated recurrent selection in finger millet. And in some cases, like in rice, people have used seedling marker like the chlorophyll deficient markers like albino or virescent, a different kind of deficient mutants they have used as a marker in commercial seed production of rice, and also in tomato they used these markers to improve the purity of the hybrids. So since we have these resources, PS1 with seedling marker, these could be utilized for commercial seed production in finger millet also. and also in recurrent selection, which was unimaginable before in the finger millet, but it is possible in now after getting this useful resources. So SNPs which we have identified can be validated. It can be validated in future days using suitable mapping populations. Finally, I acknowledge Project Coordinating Unit Millets, AICSMIP, Bangalore for supporting me in all the cases like resources, land, and in all the cases. I am very thankful to PC Unit Bangalore, and I am very thankful to ICRISAT for providing me molecular facilities and statistical data analysis facility for my PhD research work. Thank you very much for kind patience. Thank you.
Manjappa, thanks a lot. You're keeping time. Huh? That's been really nice. So let's open the floor for questions, answer queries. Starting problem, my question. Do you have any idea of linkages between virescence and sterility? How far they are linked or not linked?
Not studied, sir, but there is no linkage actually. What we see in the field, we have found both the lines having both sterile as a separate and virescent as a separate. Independently, it has seen, but specifically, we have not checked that because virescent is from different donor.
How do you see the utility of this partial sterile mutant in development of commercial hybrids?
Yeah, as I already shown in one slide, the hybrid recovery is as we seen is up to 93%. That means to say the remaining 7% are sterile. In that condition, if you grow that plants in the feed, like in the formal feed, since we are using virescent as a female parent, so when we grow under large field condition, under that condition, the plant which is self will suppress automatically, because these virescent plants at seedling stage having very poor seedling vigor, so automatically it will suppress by the true F1s, by vigorness of the hybrids, so it will not be any problem.
As this, I feel this itself is an advantage, because since it is the self one will be a virescent, farmer can remove that. As a result, what we will have is only hybrids.
Commercially, we are not thinking as of now, but this can be think of later.
If the extent of heterosis is more, it will have, if it can provide at least 10% increase in the yield levels.
MVC, sir?
This is a very nice question. We have been thinking about this particular aspect from the beginning. Say, for example, in rice, what has happened? Rice now, earlier people never thought that there could be hybrids in rice because of its reproductive strategies. Similarly, but work has been going on using even genetic male sterility since about 35 to 40 years. And a lot of information has been generated in terms of outcrossing, in terms of what should be the strategy to recover more hybrids, etc. Once the cytoplasmic genetic menstrual line was available, all that information was really useful in coming up with commercially viable hybrids. So same scenario we can think of. It may not be right now, today, it may not be feasible with this, though marker we can talk about, though transplanting we can talk about, etc. But very good commercial strategy may not be possible. But all of a sudden, if you get a cytoplasmic genetic menstrual system, tomorrow I may not be able to produce an hybrid. Okay, a lot of information is required. This system can really provide scope for doing such fundamental research to go ahead. That's one thing. But more important in finger millet is the sort of recurrent selection programs are essential, and it's very difficult for crossing, as you said, mentioned. And this would be a wonderful system one can really think about. In fact, in rice, for example, in South America, CETA, especially CET-based breeding work both in America, so Southern America, Central America, as well as in Africa, millet sterile systems have been used through recurrent selection programs. Fantastic varieties have been developed. So one can really see that sort of an application also. So I think it paves way for these approaches. Thank you.
I've seen your presentation in ANOVA. You are doing your analysis of variants as a combined one.
Yeah.
Am I right?
Yes.
So if you are doing combined one, and most of your discussion is going for season wise, any reason behind that? Most of your discussion, you are specified Kharif and summer like that.
Yeah.
But your analysis is combined. So after analysis of variance, you got something to be discussed season wise. Maybe I want to know the reason.
Yeah, sir. We wanted to see, because finger millet is growing in both seasons, in the Karnataka and in some parts of the India. In that condition, we wanted to see the performance of a hybrid in summer and Kharif. If a particular hybrid is expressed same heterosis in both the seasons, that means that that hybrid can be suitable for both the hybrids. It may not be released now, or we cannot be used now, but that information is definitely useful in finger millet. If that hybrid is specifically expressed only in Kharif or in summer, so that can be used for a specific season. So that is important in the, because of that only we have evaluated in 2 seasons.
Why did not you give you season-wise ANOVA? Since
we wanted to see the environmental interactions. Yeah, in environment interaction is more. As I shown in the ANOVA table, interaction is there for all the characters. Yeah, here you can see. For all the characters, environmental component interaction is there. Significant interaction is there. When you look at the entry level and parent level, for many of the characters, we have the interaction. So you wanted to see this. That's why you have pulled. That is heritability. That is based on 2 seasons, individual season, separately.
Govind, I will add on that. It's not about even interactions. It's actually, this information is such basic information for us to see that for finger millet, you could actually step fast your breeding program, because you can think of having 2 seasons, even for selection nurseries. You would have seen some of the slides where you would see that the correlation matrices between the 2 seasons, they are 0.7 or plus. So that's really, it will boost up. You could thinking of having multiple seasons a year, just like what you think in.
So your interaction looks more on the least side. So you can pull your data and get the stable line better. You are using the combined one. That's my question.
I would say actually more than a question, I would like just to a comment. As it was said by Santosh during the introduction, it's so nice to see this presentation on finger millet. And it's not just, I would say, a general presentation on finger millet. It's actually putting some basis for, it's an important component for bringing together the pieces for a well-structured breeding program. And that's, I think, what is the importance of this, of the outcome, of the outputs of these pieces. Of course, these are built on the large amount of knowledge that has been generated so far by the program, program in Bangalore. So it's not something like coming out of one PhD thesis, but definitely put a lot of basis that could be useful not only here in India, but I'm thinking, for example, at the application of this type of information for our finger millet breeding program in Africa. Considering that for until a few years back, actually most of the breeding work done on finger millet, at least now I'm talking about the experience in Africa, has been mainly selection from populations. That was because mainly there was no, the problem of hybridization was there, and so I think it opens all sorts of new avenues for the improvement of this crop that it has a great potential. Not only here in India, I know it's a very important crop, both for food security and nutrition, but in Africa it's an important crop. I'm not putting down per millet, don't worry, don't look at me, I know he's getting worried about per millet, but and it has also, I mean, there was an article recently, I can't remember on which journal, but a number of people are actually comparing, talking about the finger millet is the next quinoa. Now, we all know the story of quinoa. We all know how it went from being an almost unknown crop in the Andean region to being now on the shelf of almost all supermarkets around the world. Perhaps we can learn from the quinoa story and not repeat the same mistake that had happened with quinoa. But a lot of people are looking at finger millet as the next superfood that could be just the quinoa number 2. So, and definitely that information is extremely important to move in that direction. Sorry, that's all I want. So thank you very much for that, contributing to that walk of finger millet. Thank you.
So if we have no more questions or comments, let's thank Manjappa and congratulate him in usual way for his seminar. And we have a representative from HR looking after LSU business, Ms. Sriprada. She would be handing over a certificate and few more comments maybe. And before I do that, I would really like to take this opportunity to thank both Dr. M.V. Sigowda, Dr. Rangaya. Thanks very much for taking time and coming here and visiting us for this seminar at very short notice. We really appreciate it. So we hope that we would continue our collaboration, as I said again, and probably the last but not the least, thanks Stefania for your time and all your persuasion to make addition of one more crop to our portfolio. So I could still keep my love for finger millet intact officially, not to justify what I work on finger millet between the lines. So I can put that.
If I can correct you just slightly.
Yeah.
Actually, because that's also the message that our DG is always conveying. So at ICRISAT, finger millet was always one of our crop because it was part of the small millet group. So what it was actually decided, what the governing board decided in the last meeting was actually to make it more visible, taking a finger millet out of the small millet that we still have with us, but taking it next to the other crops. So, but definitely is, for me, it's also an important step. Thank you.
Thank you very much.